what I'm going to be talking about today mainly is the field that's emerged, really begun flourishing only over the past 5, at the most 10 years in the way you define it. And one of the aspects that's so complex about this field is that it's, and which I'll try to argue, is that it's been 2 parallel lineages that linked up around 2003. And so it's 2 parallel stories, 2 parallel research agendas, sets of researchers that have fluctuated between various fields, various disciplines, the types of questions that they asked, and the terminologies themselves have become sort of conflated. So it becomes difficult to differentiate them, to understand what's going on, to understand what the motivations are behind these studies, and also difficult to tell this story, because you have to tell these 2 stories in parallel. And I'll do my best to do that, but hopefully we'll be able to interact and let me know if there are questions as we move along. And then finally, towards the end, I'll present some of the future directions, where the field is at the moment, and where it's looking towards. So this is really where it all began. In both the stories that I'm going to tell, this is the symbol that one could use if one were to create a logo. It's your basic crosshair from a cognitive neuroscience experiment. And of course, while it comes with all of the other kind of cultural, symbolic aspects that a cross does come with, this is the symbol that usually is put up as a stimulus presentation when you want someone just to relax, to establish what's called a baseline. Something that doesn't say, offer anything towards the specific question that's being asked in a psychological sense, but keeps the attention of the person focused and ready for what's going to appear next. What happened in both of these fields is that the question became, rather than looking at what was evoked through the stimulus, became what is happening in the brain during this baseline condition. What is taking place, both in terms of the spontaneous internal mentations that one might have, but also what's happening in contrast to the kinds of activities that we're interested in a task-evoked state. And what's been remarkable over the past decade in looking at data that's been acquired during the specific paradigm is the degree to which it's similar both across individuals, the degree to which it reflects underlying functional organization of the brain, and the ways in which that can be used, even independent of all these complex manipulations that are used, to be able to understand brain function. And so I'll go into a few of the methods that are used to do that. But essentially, this has really been, until a few years ago, this dark side of cognition, this side that we couldn't understand what's happening during the baseline state. This is the part that couldn't be probed. This is the part that we weren't interested in. Now the question has come up methodologically, how do we begin to address it? Just to try and reinforce the element of the unknown that's present in this idea of a crosshair, in terms of popular press, the metaphors that are used to describe this new eruption of interest in what's going on in the baseline of brain activity, terms that are being used in the popular press, this is psychology's very own dark matter that comes with all the implications from physics of the excitement, the potential for understanding, and also the great unknown of the field. It's like finding a new continent or working through uncharted territory. The way in which this is being portrayed in the popular press, which again I think is largely due to many of the prominent researchers within the field, is mapping it as a place of discovery, a place of the unknown, and essentially a paradigm shift in the field. Now, whether or not this is the case, I think that is definitely something that can be put up for debate. There are many reasons why it doesn't fit what would be traditionally considered a Kuhnian paradigm shift. However, for the sake of this, what I'd like to focus on is the fact that researchers are titling their papers a paradigm shift in functional brain imaging, and that folks like Marcus Rakel, who's one of the prominent players in bringing this platform to the research community and to the kinds of questions that are being asked in the research community and defining those questions, defining those interpretations, is portraying this as a dramatic shift in the way that people are understanding brain activity, the types of tools that are being used, and offering a completely new landscape, to continue with the kind of territory metaphors of what can be done in the field now. So as soon as we begin talking about brain states during fixation on a crosshair, during a state which is not being controlled by the psychologist, it immediately moves into psychological territory that would be described as mind-wandering. And that's precisely what the field has done in terms of reaching from the brain sciences in which it developed into the psychological and cognitive territories that it's been gaining, say, a popular consciousness. So the kinds of questions that are being asked in the field, the kinds of implications that it's having are for what is the value of mind wandering, what is the value of resting activity for creativity. And as you can see just from some of the headlines that have appeared in the popular press, the implications of resting state research, in part due only to the term itself of resting state, are vast in terms of thinking about the value of, say, this mental state for productivity and for cognitive life. So a couple of the guiding questions here, and this is where we'll begin to go through the division that takes place. One of the initial questions that launched the field is, what is the cognitive baseline state of the brain? So in a sense, what is going on in the brain, but also in mental life in these areas in this baseline state? And that will lead to one agenda that will be described as the default mode network agenda. Another one is, what is the physiological baseline? So looking at spontaneous brain activity, in this case, what makes up the bold signal that we just learned about in the previous lecture? What constitutes that signal? What are the various aspects, both physiological and neuronal? What is the functional meaning of it? But really, what is the physiological baseline if we want to be comparing this to something that's, say, evoked through a specific task? And then in large part, what these lead to is the question of what is basic brain organization? And so the techniques that have been developed in these two, specifically in this second one, are going to lead to applications for understanding and mapping the connectome, which is one of the most prominent, I would argue, prominent research agendas that we have set forward in neuroimaging for the next decade. So here's the basic timeline that I'll be working with. And the reason this is divided between default mode and resting state is because, as I mentioned, these both have very unique historical origins. The questions that they're asking are different, and even the methodologies that they're using are different. But from a certain point onward, they're going to merge, and all sorts of problems arise when that happens. So let's start back at 1997 with the default mode half of this. To begin with, just a very basic review again, as we already went through, but just to give it for the context of this story, this is your standard paradigm, just for the sake of having a model of it. You're going to have one task, say one state, it could be finger tapping, and then an off state. In this case, we'll call it a resting baseline with just a crosshair fixation. So every 20 seconds, you have this crosshair that comes on. It says, just stay there, do nothing. And then for 20 seconds, a little, say, F will come up that tells you to tap your fingers. And so you're tapping your fingers for 20 seconds, you're not for 20 seconds, back and forth and back and forth. You subtract one state from the other, and you can see areas of the brain that were more active when you were tapping your fingers. What's often been used in this case is having a resting baseline. But for, say, the first decade of imaging, from 1990 towards the end of the 90s, the question of what was happening in this resting state was not something that was specifically probed. It was simply used as a neutral state. It was necessary, it is necessary, to have a contrasted state in order to investigate any activity. And so rest took that place. But what happened that launched this field in 97 was a meta-analysis that was conducted by Shulman and colleagues at Washington University, working with Mark Rakel, who was mentioned already for announcing the paradigm shift in imaging. What he found was that by doing a meta-analysis of PET studies, you could see a consistent set of regions that were more active during the baseline state than they were at rest. Now, it's not like these had magically appeared. People had noted them. They just didn't know how to describe them. Because when you're talking about contrasting a state that you're interested in with one that you're not interested in, it's rather awkward if all of a sudden you've got areas of the brain that are more active in the state that you're not interested in, because you don't even know how to talk about it. It's an unconstrained state. It's a baseline resting state. So people had focused on the one they were interested in, and finally, 97, a second paper in a two-part series comes out by Shulman where they look at this meta-analysis and find we have this consistent set of regions. We don't know what they're doing. All we know is that across various studies ranging from visual paradigms to different sort of memory paradigms to word generation tasks, there's a consistent set of regions that are active in the resting aspect of that. What could they be doing? And it takes another few years for them to begin to develop that question. But it was simply, Felicity Collard, the co-author of the article that's in the packet, and I affectionately term it the flipping of the contrasts as a historical moment. And that's all it was. It was simply looking at one over the other. No major methodological difference except to focus attention on what was less constrained, what hadn't been the focus of these previous studies. a few years later, in 2001, 3 articles come out, all co-authored by Marcus Raichle and the Washington University group, in which they begin to assign a term to describe this state of brain function, this state of psychological life. A default mode is the term they use. And this harps back, of course, to computer terminology in terms of your standard state of operating. But they ask the question, what is the psychological state that we could call default mode? And what are the brain regions that are more active doing? What are they specifically responsible for? What are their functions? And how can we study it if we're, up till now, have been studying it in this unconstrained state? What they begin to ascribe to it is a self-referential role, an episodic memory role, and there are other reasons that I can go into for why they ascribe these specific functions to it. But this launches around 2001 the question of how do we understand the default mode of brain function. This is not the baseline state that we'd hoped that it was, that we've been using for a contrast. Things are happening. We're all aware. If I put that crosshair back up and have everybody just stare at it for a couple of minutes, your minds don't go blank. There's all sorts of things that you would be thinking about, whether it's about the future, what you might be doing for lunch, whether you should be working on this or that, or our minds are wandering. There's specific psychological content to that, and that content has associated brain activity, which is a confound if you're using it as a contrast state. So the question came up that if there is this activity, we can't be using this as baseline. What is it that we've been using as baseline, and how can we understand it? I want to pull up this paper because this has been recently, the history of this field is still being written and rewritten. If anyone is interested in going in more deeply to the history, Neuroimage, one of the journals that's prominent in publishing papers from the field, just put out a series of, I think, half a dozen articles of people who were involved in the origins, actually of fMRI in general. And people were able to write first-person accounts of what was going on in their labs, how they were involved, essentially writing their own histories of the field. It's a wonderful set of articles because it gives you a very personal insight into the power plays that were taking place, how people envisioned themselves as taking part in those initial discoveries, and what the discoveries themselves were, what made it an innovative time to be involved in the field. But one of the papers that was brought back historically as being somehow at the root of our understanding, or at least the first time that people really thought about what rest might be doing and termed it as such, was in this paper first authored by Nancy Andreasen, in which they term rest random episodic silent thinking, and give it the acronym REST, which I think is cute, to say the least. What they did was to look at various types of memory. So in one case, episodic memory, in another case, semantic. And as you can see, if you're contrasting episodic memory minus rest, it's hard to see from this, but the blue regions, which are the regions more active in the rest state, aren't the ones that we would traditionally associate with the default mode, with those regions that I initially showed you. But if you look at the semantic memory versus rest, the regions that would be implicated in the default mode traditionally are higher in activity during rest. And part of what this goes to show is the bias in the way that we understand the default mode is based on the interest of researchers in the types of tasks that they were studying. So I'll say that again. If people had been interested in the kinds of brain activities that we associate now with the default mode network, things like self-related processing, like episodic memory, and they had contrasted that with a resting state, you wouldn't have seen the default mode, the areas that we now consider to be the default mode areas. What's become the default mode network in large part is due to the interests of researchers within the field in the 90s, and it essentially sums it up. Another aspect about this paper that I find interesting is that because there was description of areas that were more active at rest, it was necessary to give it this very complex acronym to describe it as a cognitive state. So although it's essentially telling people, just hang out and think about yourself and your own past, things that we would naturally do while mind wandering, it was necessary to term it as such because it had to be described as cognitive state that had certain areas associated with it. So it seems like in part we're starting to get closer to understanding the function of the default mode, and then all of a sudden last year a paper comes out that shows that rats too have a default mode network, which makes things very complicated in terms of trying to understand what the functional role of this might be. It had already been shown in 2007 that monkeys had it. It's been shown that these areas are a network when you're under anesthesia, when you're asleep. In I think chimpanzees, it's also been shown, in macaque monkeys here, and finally in rats, which means that the ideas of this being implicated in some sort of self-related processing or episodic memory, as we understand it, are probably not the case here. It raises problems for interpretation, except, and this is where I have to put a really strong warning on this, the techniques that were used to extract out these networks are very different from the ones that you previously saw, in which you had two different cognitive states, either resting baseline and an active one, or one that was involved in, say, some self-related processing measure and one that wasn't. The techniques that are used here to pull out these coherent regions are based on correlations of spontaneous activity. And so it has nothing to do with the cognitive state. The fact that you have coherence between these regions could be something that's phylogenetically preserved, but functionally is irrelevant in terms of the way we think of its use in humans. So it gets more complicated. And this is where I need to jump now a step back to the other story that's led up to this point, which is one of looking at spontaneous brain activity and functional connectivity. So let's say by the time we get to 2001, we've situated this question of the default mode of brain function, the idea that there are certain areas of the brain that are more active in a resting state, and the question of what those might be doing. How do we study this task-independent state? It likes to do that. 1995. A paper comes out in 1995 that essentially launches this field. And just to give you a feel for what the questions that initiated this paper were, I'm just going to show you here. This is fMRI data as it would come off the scanner. And I've colorized it so that red areas are higher in activity, blue areas are lower in activity, and all I've done to this data is subtract out the mean values so that you can see it on this nice little kind of color scale. But the one other thing that I've done is to smooth it in time. So normally you would be acquiring a volume every 2 seconds. In this case, I just smoothed it so that it transitions more gracefully. But what you're seeing here are the kind of fluctuations that we're dealing with in fMRI data in real time, in the sense that this is how they come off the scanner. You can begin to see, even just by looking at this, that there might be areas that seem to be rising in activity in synchrony, but that there's also a lot of noise present. And so the challenge that, so just to put this in context, the challenge that we're faced with as a field now is trying to discern specific networks of activity that are coherently present in this data, how to essentially be able to analyze this data, to understand what areas are talking to each other and what that might mean. But the question that kicked this off was back in 95, and this was a researcher, Bharat Biswal, who was working with James Hyde at the time in Wisconsin. The basic problem was that if you've got bold signal, there's all sorts of things that play into that, as we already saw. This is a blood response to neuronal activity. And if you take, let's say, one voxel of the signal and you transform it into the frequency domain, so you look at what frequencies make that up, you begin to see specific peaks that are present, which means that the signal in these areas, that frequency, is more present in the bold data that you're looking at. And while they were able to call that cardiac signal there around 1 hertz, call this respiratory activity, one of the big questions that was posed around 95 is, what is all of this low frequency activity that's taking place? Why is it there? What's it doing? Because as far as we know, bold signal without some sort of modulation of brain activity, without some thing that's waking you up and telling you to do something, without a little F that tells you to tap your finger, there's no neuronal activity that should be of interest at that point. It's just noise. And so that's the question that Bharat Biswal started with. What is this low frequency activity that's unaccountable for by physiological information? I'll take you through the methods of the paper because it's the basis for this agenda of functional connectivity. What he did was to take people, put them in the scanner, have them do the finger tapping task that I just described. And so just for the sake of it, I'll model it like this, but let's say that this is where you have finger tapping on and off, and what the bold signal response should be to that activity. And as you can see very nicely, when you run a finger tapping task and you properly analyze it, you get areas of the motor cortex implicated in that activity. So far, no big surprise. But then comes that question of what is going on with this spontaneous section in the very low frequency range. And so what Bharat did was to take a region of interest from inside the motor cortex, an area that he knew to be involved in the tapping, extracted out that time series, so simply took the spontaneous data from this area as it was acquired at rest. And so here's an example of a time series from that area. Looked at only the low frequencies that we saw before, so simply cut out that little section at the end to say what might this set of frequencies be responsible for. And when you look to see what was correlated with that, so spontaneously, independent of any task, what areas of the brain were spontaneously correlated with activity from this area in the motor cortex, he found the entire motor cortex to be correlated at rest. This was a major revolution in the field because it meant that what we were considering noise, what we were considering to have no functional significance, what we thought could only be coherent when you were asking the brain to do something, was somehow also organized spontaneously in low frequencies at rest in the bull data. So this major disadvantage that I think everyone can agree with, of fMRI having a very low sort of sweet spot for the frequencies that it can acquire, turned out to be a major advantage when it came to looking at these coherent networks at rest. I'll just show that in another form, but this is the exact same message, which is that if you take the right and left motor cortex and you look at its spontaneous activity at rest, it's highly correlated. Somehow functional systems, at least in the case of primary motor here, appear to be organized spontaneously. So the question of what is the physiological baseline, what is the neuronal baseline, there appeared to be a functional meaning to spontaneous brain activity in this case. On a more of a historical note at this point, it took surprisingly about a decade for people to make use of this finding beyond primary sensory areas. And there's a researcher by the name of Randy Buckner who's done a lot of work with memory, prominent cognitive neuroimager. When he first began to see this work coming out, he was very, in a sense, disappointed in himself because nobody could think far enough outside the box at that point to realize that something that seems so self-evident today, that all you have to do is look at another brain region and perhaps you would see another network that was meaningful. People couldn't even conceive of that at the time because it was such an odd finding within the context of how people understood brain activity from a cognitive psychology perspective, which was the prominent perspective brought into neuroimaging. But so around 2006, you begin to see people analyzing the data with a new set of tools. They're using something called independent component analysis. It allows them to throw in a data set and see what comes out, essentially, what are the independent networks. And they begin to find all of these networks that are coherent at rest. All these networks that they've been seeing for decades in cognitive neuroimaging are beginning to arise again at rest, independent of any specific task. This is simply showing two different methods that are capable of deriving the same networks. But these are networks that if you were to go back to 95, or say a little bit later, end of the 90s, and show to cognitive neuroscientists, they would be able to identify them from the task domain. And it was, in essence, that task domain that gave meaning to these networks, because you could begin to ascribe function to them. So at this point, Jan loves it when I show his brain in class. So I felt that it would be best just to give you a quick feel for one of the tools that can be used to understand, to look at functional connectivity and how that might work. This is Jan's brain. Thank you, Jan, for we've cleared all this in consent forms ahead of time. And I apologize, there was a little technical problem that I ran into as I was setting this up, so it's not perfect, but I just wanted to give you a feel for the software, one of the softwares that can be used. In terms of, again, just a historical note, this software, which has this wonderful kind of 80s cockpit field, was developed in the same lab in which Bharat Biswal was working back in the early 90s. So it's one of the softwares that's been most advanced in terms of the methods that can be implemented for looking at resting state data. It's called AFNI. And if you pick any part of the brain and simply click on it, it allows you to very quickly, almost instantaneously, visualize the other areas that are correlated with the activity from that seed region, as it's termed. This was released I think New Year's Eve 2009 is a present to the community from the creators. And so it's just a matter of clicking anywhere and, in theory, seeing the areas that are spontaneously correlated with that. This brings the question, just to go back now, this brings the question very far away from the psychological one of what is happening in the baseline state. This turns it completely into an anatomical question at this point. What areas are functionally connected to one another? But essentially, there are tons of techniques, and I doubt that this is legible from the back. No, kind of. But there are so many techniques that have arisen for addressing spontaneous brain activity, essentially because you've got many different ways to think about it. You're dealing with all of the tools that you could pull from signal processing to look at time series. And then once you take those time series and you somehow relate them into a correlation matrix, so you look at how each one relates to every other and you can just plot it as a relationship between all of them, then you can move into the field of mathematics that's called graph theory and begin to talk about these as graphs. So that there are all these different tools, all these different fields that have sort of jumped on the excitement of this data, what can be done with it. The problem, of course, begins then, what is the question that you're asking with the data? And this is where people have had to take a step back again and not just throw their data through every single toolbox, but to begin to ask more specific questions in terms of trying to understand functional connectivity. One of the researchers, I think, that's been doing a wonderful job of pushing the field forward is, his name is Steve Smith. He's at Oxford. And every year or so, he comes out with another paper that overturns a lot of the conceptions of the field, asks a new question, motivates people to begin really thinking about what we can do with spontaneous brain activity in a different way. This is one of those papers. It came out in 2009 in PNAS. And what it did was to demonstrate the relationship between the networks that were seen at rest and the functional imaging literature, so the networks that have been shown through task-induced activations, and that the two corresponded. And why this was important, I'll show here. So first, what you're seeing here, Smith and colleagues took a database that's called the BrainMap database. You can access it online. It's about 9,000 data points across hundreds of studies, I think dozens, hundreds of studies, where they're looking at where you see activations during various tasks. And if you conduct a meta-analysis of that and then decompose it using ICA, independent component analysis, essentially what you end up with at the end is a set of networks that are related to these various tasks. These are things that co-activate together. So you can break it down into a set of coherent regions across these tasks. That's the task aspect of it, and that's what you see on the right side of each of these images. On the left side, what you're seeing is a similar kind of analysis on resting state data from 36 people. And what this is showing is a correspondence between the kinds of patterns that you get from the task data and the kinds of patterns that you get from 36 people at rest. If you look across the different task domains, and here you're seeing task domain over the different networks, and you look to see how each one of these gets divided up into the task domain, it's not a clear relationship. And I think that's really the crucial finding of this paper. But that there's something more basic about these network structures and the fact that these are coherent both in the task and at rest that's not playing out in the kinds of psychological categories that we would traditionally use to study them. This has been termed dealing with what's called a functional ontology, so the way in which we understand the basis of these various functional categories. And as they've been extracted from cognitive psychology, which was a pretty good guess at what's underlying the basis of brain function, but as you can see, it doesn't map one-to-one to the actual networks that are present in the brain. And it raises the question of where should we be starting with if we're trying to break up the brain to various functional modules. The main argument of this paper being that resting state is a pretty good way to start, or at least spontaneous brain activity. Yeah, please. So let me, given this discussion, let me try and reformulate what the importance of this shift is. The notion of a one-to-one correspondence between some psychological function and a brain area is kind of the dream of cognitive neuroscience. that you conceive of some construct, and you're able to map where it's happening in the brain, and that it's a clean module that's responsible for that function. What this demonstrates, this paper demonstrated, is that given all of the data that's been acquired over the past decade that's been mapped in this database, there is not a one-to-one correspondence between them. Yeah, and so, however, for whatever strange reason, the kinds of co-activation patterns that you're seeing in the task literature are also present in this resting state data. There's something about brain organization independent of a task that's also capturing these, and I'll use the word coherent because of spontaneous correlation, but these correlated networks, these networks that are correlated in their spontaneous brain activity. It gives hope for how, at least to the field, for how this could be used to really study brain organization, which could then be mapped back in terms of starting with this and then trying to understand the constructs rather than taking the constructs and trying to understand the modules in the brain. That's the basic shift that this paper proposed. Does that help? Does it? Thank you for asking. Okay, so back to the timeline, catching up with the timeline. 1995, we've got the start of this resting state functional connectivity. Again, a different question of what is physiological noise. How can it be used to understand connectivity between networks and the presence of these networks independent of a stimulus? In 2003, this is the watershed paper for the whole field, because this is the one that brings them together for the first time. It's a paper by Michael Grecius and colleagues who were working at Stanford at the time, and still are. And it's the first one that cites both the Biswal literature and the Reichel literature in the same paper. And even the title itself reflects the linking up of these two research interests. So functional connectivity in the resting brain, that's a shout out to Biswal and colleagues. A network analysis of the default mode hypothesis. And the default mode hypothesis, again, is this idea that you have a coherent set of brain regions that are responsible for baseline, say, psychological states during this resting task. They did something very simple. They took the Biswal approach of putting a region of interest in the motor cortex, and instead they shifted it to an area that was within this default mode network that had been found by Reichel and colleagues before. So they just looked at an area of the brain called the posterior cingulate cortex, what they saw were all these areas of the brain that had been involved in the default mode were correlated at rest. And it was a way of linking up these two literatures, both around the term of resting state, because both of them to some extent were interested in this baseline resting state, though with very different questions. Also around these areas, around the idea of functional connectivity, began to confuse things. So it raises the question now, what is default mode? What's a default mode network? What's a resting state network? How does this differentiate from the default mode? What are the defining features of all of these? To the point that you today, reviewing papers, even in the literature, you see a lot of confusion from researchers in the field in terms of defining these various terms. This is also the first mention of the default mode network as such, rather than being the default mode as a brain state. This is the first one that says that this constellation of regions here can be referred to as a network rather than as the baseline state. And that's a crucial shift, because then later on it allows for one to have a paper that says rats also have a default mode network without in any way addressing the psychological state that's associated with it from its original definition. One can simply look at the coherence of these brain regions and describe it independently of any psychological definition. So if this seems confusing, I assure you it's because it is. It's a mess, the literature in the field right now. It's bringing together many different literatures and using definitions quite loosely, I think, to the detriment of having a better understanding of, yeah. Okay. So it's an interesting point that you're bringing up, because I think the initial papers in 2001 were actually counterintuitively trying to do exactly what you're describing, which is focus on a state that had just been assumed to be a default, to be a neutral state, and begin to investigate what's actually happening in them psychologically. The problem was that the term that was given was default mode, which carries with it the meaning and the, say, assumption that move forward with the field of this actually is somehow a default mode of function, rather than this is what we've considered a default mode that needs to be studied further. Absolutely. And that was part of the biases of the cognitive psychology that was brought into as the basis for cognitive neuroimaging work. Let's jump back to these two different approaches, these two different types of questions. For the Biswal stuff, to be perfectly frank, the question of what someone's thinking about is not as important as simply looking at what is happening in the bold signal and is there neuronal activity underlying it that might have some functional significance. He was looking at finger tapping in this case, motor cortex. Even if someone's imagining themselves playing tennis, it's not as important as understanding that there's meaning to the low frequency activity in the bold signal. And then it turns out that these same fluctuations are tying together a lot more functionally. For the other side of it, that's precisely what they were criticizing. And I think you're very much aligned with what Rakel and his group in 2000 were frustrated by, that we're calling this thing a resting baseline condition when it's not. And as it is not now, let's try and investigate what actually is going on and how can we continue to do that. So they're very much in line with your way of thinking. Now, what's happened after that is a bit messier. And that's where certain, because of the terms that were used, because default mode was the name that was given to it, again, like against probably the hopes of the initial researchers who conceived of that term, this has become used as a neutral kind of state. We can just throw someone in the scanner and then get all this information about their brain. It is problematic, and there is work that's being done to try and address state-dependent differences in activity. Now, and to extend your metaphor, this is the equivalent of letting the computer continue running and then looking at how things are communicating within it when you're not running a specific program. So you just have it up and going, you haven't turned on Microsoft Word yet, but you start looking at what's talking to what. And then the way it's been done in the past is you turn on Word, you see what's involved with getting Word running, and then you can know, okay, this is the part of the computer that does Microsoft Word, for instance, or whatever it might be. The individuals that were involved in really the birth of cognitive neuroscience meets these scanners, folks at UCL were virulently against resting state. There were several papers. I think one of them had the subtitle "Resisting Arrest," which is cute. Why we should be resisting arrest? Why we should be resisting arrest by Morcombe and Fletcher. There was an enormous amount of frustration for this resurgence of an interest in trying to study brain activity without actually controlling the cognitive state. And why on earth would you try possibly to understand function that way? And so one of the counter-arguments to that, which then actually, this is what happens, Rakel then takes, just to continue if it's okay, and if there are other questions, please. But what happens at this point is that you see the merging of these fields, and this actually bridges right back. So all of a sudden now, Rakel, who's been hitting this kind of dead end with the idea of looking at the default mode of brain function, in the sense that people said either we got to study this in task, or we're not studying these brain regions. There's no way to do it. You can't study the areas of the default mode network in a baseline state. You have to do tasks that you think would implicate it. All of a sudden, Grecius is able to show in 2003 that these areas are correlated at rest. So you've got rest baseline on the one hand, which is the cognitive state. You've got resting state connectivity on the other hand that shows these to be correlated with each other. Perhaps one way to unify these fields is by rethinking how we understand brain function and just simply shifting the agenda of the entire field as Reichel conceives of it. And so they call back to a book. This is in the introduction of a book by Rodolfo Llinás, but where they talk about these two major modes of thinking about brain function, one in which the brain is primarily concerned with managing input and output of stimuli. So it's this classic idea if you have the black box, cognitive psychology, you've got an input, something happens to deal with that input, and you have an output, and that's what's important to the brain. It's more important what is happening in the outside world. And then you have this other model of brain function, which historically goes back 150 years at least, in terms of spontaneous dynamics in the brain, and that quite frankly, the prefrontal cortex is more interested in what this part of the brain is doing than indirectly what's happening out there in the world. So that, which, I mean, if I were the prefrontal, I would feel that way, I guess. Yeah. But the idea of the environment is modulating the internal dynamics in dialogue, conversation, interaction, information transfer that's happening in the brain rather than determining it in terms of an input-output machine. And this is where Reichel positions himself in terms of understanding the value of moving forward with this research agenda. It's about a prioritization, a reprioritization of what's historically been always present, this interest in intrinsic dynamics in the brain. Not conceiving of the brain as an input-output machine, but having its own dynamics that are involved with its self-organization. These are always present. The fluctuations that we thought were noise are actually ways that we can understand brain organization. And rest just happens to be a state that facilitates studying that, but it's not necessary in order to be looking at these spontaneous dynamics. Okay, so just to wrap things up in terms of where this is going, I put this up because it was alluded to yesterday, but the connectome has become a very hot agenda in what can the potential applications of neuroimaging and these technologies be. $40 million were just given by the National Institutes of Health to develop both the tools as well as provide the data to study the human connectome. This is one image that was created by a postdoc in my lab that was meant simply to look very impressive. I'm really happy with him about that.
40 million tags.
I'm sorry?
40 million tags.
Exactly. I wish we'd benefit from it. But this goes back to 93. At least this is one of the earlier big outcries for how can we be studying the human brain when we really know so little about its organization. And this was a paper by Francis Crick and Ed Jones. And Crick, you might remember from DNA fame, who also, he has a deal with Nature where back in the day, he could send them anything, they would print it for him. Yeah. And so in this one, he's doing a 2-page shout out to, we have got to try and understand the totality of connections in the human brain in order to be able to talk about its function. And it is intolerable that we do not have this information referring to connective information for the human brain. Without it, there's little hope of understanding how our brains work except in the crudest way. And so the object of this paper is to make this more widely known. So this just gives a sense for this is 93 when this is happening. Functional neuroimaging is just about to emerge. He even makes certain points in the paper that within years are going to be addressed by fMRI back in 93. The field's on the cusp of it when this comes out. And here we are now with the NIH finally giving the money, putting forward the call for proposals and giving the money to a couple of institutions to begin addressing these questions on the scale that the Human Genome Project took place. One day we might have a picture in our wallets of our connectomes. This is some work that recently came out trying to look at ways to do multimodal visualization of the full connectivity map of the brain. Again, I'm putting these up to show the direction that people are thinking in with conceiving of brain organization on an individual level. This is one person. This is not a group level map. So while a lot of the past 10 years was thinking about ways statistically to be able to look at group level statistics, group level functions, now the methods are pushing back towards how we can think of this on an individual level and how we can use many different modalities to say understand this complex organization. And while this certainly contains a lot of information, from first glance, it certainly doesn't help with the immediate comprehension of brain organization. Exactly. I simply put this up. Van Vedeen has been a major innovator in the field. His most recent work is trying to come up with very basic principles to how wiring takes place in the brain. And in this paper that just came out, it's one of these kind of revolutionary perspectives that tries to dramatically simplify our understanding of the basic organization of wiring in the mammalian cortex. This is simply one part. The technologies, I won't go into detail now, but there were new technologies that were innovated in terms of scanning to be able to even address those, do that kind of imaging. But they are part of this Human Connectome project. And this, what you're seeing on top, the logo, the pretty website, the nice graphic design is all part of this $30 million that were given to WashU, and then another 10 that went between UCLA and Harvard to have a comprehensive effort to map brain connectivity and its variability in over 1,000 healthy adult subjects, producing a large amount of current. It's an incredible project. For someone in the field like myself, when this data set's released, none of us are going to have to acquire data for a long time. They're putting out tools. They're putting out fancy logos. I would be surprised if they don't have a little jingle by the end of this. It's really a remarkable, the quality of data that they're trying to achieve in terms of twin data acquisitions across several different modalities, numerous cognitive tests, and phenotypic information is being acquired. It's going to fundamentally change the field in 5 years when this data is released. Because anybody in this room could decide they want to get into this field, download the data, download the tools, and start writing papers on it. I think that's a question that people are absolutely right now struggling with, the parallels with the Genome Project, and if we're falling into the same pitfalls. I'm ready in 10 years to start looking for a new field. But on the other side, that was more of a joke, but the other side of it is there's a big difference between the Genome Project and the kind of data that was acquired and what's happening here, which means I think it's going to be more difficult to see when this has failed than the Genome Project not playing out the way people would hope. And in more detail, what I mean by that is that the kind of data that's being acquired is not really the neuron-to-neuron connectome. It's these kind of complex, non-invasive imaging techniques to try and describe, through various modalities and different analyses, the connections in the human brain. But it's not actually the information that we're claiming to describe itself, which means that there's a lot more flexibility in how it gets analyzed and the way the results can be described. One of the major goals of this project, and I'll jump to the next slide, for instance, disease prediction. Predicting variations in cognitive phenotypes is going to be a major goal of this project as it's put forward now. So this is a funny example, and let me just mention the body, if you don't mind, and we can jump back and forth, but I think this is kind of I thought this would be fun for everybody to think about together. So the top half, that's the Microsoft version of trying to understand the connectome, in that they're still in the data acquisition phase right now. They're putting out papers that are describing the protocols. It's going to be a long time before this data is packaged and ready. And it's one institution that's behind it, essentially, that's received the money. The bottom, this is the Linux version of the Human Connectome Project, 1,000 functional connectomes. It was basically a few folks at New York University who were able to convince the community to put their data into a big pool and make it publicly available. So it began with around 1,400 data sets. And this afternoon, if you feel so inspired, you can go home and download this data and the tools for free and get going on analyzing your very own resting state, as well as other modalities of imaging data. The implications for the bottom one, in terms of what is considered now valuable currency in the field, are important, because it's no longer the biggest imaging facility that can get the hottest new data. Everybody has access to the data now. We all have access to this data now. There are no excuses for not having data. So now what becomes valuable is the ways in which people are thinking about analyzing it. And it's simply changed publication in the field over the past couple of years. Like that. In fact, I would like to actually say that a lot of the presentation is reflecting perspectives within the field rather than my own. But I'm trying to do my best to voice those, because I think the way people are considering this right now within the field is pretty interesting to the critical neuroscience agenda. So I just want to say that I completely respect how controversial that is, and as I was making that title, but I'm putting it up. Again, I didn't know what to do. Like, typically developing children, do you take it out? Do you say, like, healthy controls is also problematic? So I just decided to go straight from the website on these. Very quickly, what happened, 800 or so, how many? Yeah, around 800 data sets were released across 8 institutions. The idea being, here are 800 data sets. Can you tell me who has ADHD in one of the 2 subtypes, according to the DSM-4TR, or not? And what is your prediction accuracy according to the diagnostics that were already conducted on these individuals? There was the resting state data that was given out, as well as anatomical scans, sort of minimal amount of demographic information. 21 submissions were put forward, and the basic submission was a classification based on these kinds of different machine learning approaches to data. There was 60%, things were peaking at around 60% prediction accuracy, so it actually didn't go that well. The funny part about this, it was like watching a political scandal take place. So this is Tal Yarkoni, who's working with Tor Wager out in Colorado, put a blog post up after the results came in, entitled Brain-Based Prediction of ADHD, now with 100% fewer brains. And the reason that it has this title, I thought it was really fascinating. This is a quote directly from the website, and he's just contextualizing it here. The team that scored the highest in their prediction accuracy was from University of Alberta. They did not use imaging data for the prediction model. This was not consistent with the intent of the competition. They were actually excluded from the competition because they didn't use the brain data, but they did better than everybody else. So instead, they used only age, sex, handedness, and IQ. However, in doing so, they obtained the most points, outscoring the team from Johns Hopkins by 5 points, as well as obtaining the highest prediction accuracy of 62.52%. So to put it differently, if you want to predict ADHD status using the ADHD 200 data, your best bet is to not really use the ADHD 200 data, at least not the brain part of it. And watching the way that the organizers responded to this, of course, because it's kind of a political scandal at that point. We put out something trying to show how valuable prediction of brain data can be. We had the entire imaging community, in a sense, competing to see who could perform the best. And it turned out that a group could perform the best if they avoided the brain data. So it's easy to make fun of this. I'll say that. The way, of course, that it was spun is as you'd predict. People were hopeful that this is the initiation of something, that a lot of good ideas came out, and all the classic kind of rhetoric surrounding that. And I'm torn because, of course, I'm very friendly with the organizers, and I love to tease them about this, but they take it very, very personally. Yeah, so I don't know, I don't want to, I'm very conflicted in presenting this because I can see it from both perspectives, and it was just really funny. But the other side about where this is going, and this is my second to last slide. So here's the other side of this, is something with very high prediction accuracy. This was a paper published in Science in 2010 in which they developed this idea of a brain maturity index. So the same way you would go to the doctor and get your height measured, and that would be plotted along a normal distribution of heights over age. The idea here was that you take functional connectivity, which, as described in a lot of the press releases, allows you to see thousands of connections to the brain with only a very short scan, and then you can plot out where somebody falls in their normal distribution for their age of development of their brain connectivity graph. This is the kind of stuff that people are dreaming about when these data sets are being released. And I think this is this is an indication of where the field is going to be trying to go. Right now, I know that a new consortium of data is being set up for autism spectrum disorder data, and that's just going to be happening more and more. Epilepsy, there are consortiums being set up. So the idea is, one of the reasons people have been asking, why rest? Why stick to this notion that cognitively we know to be so invalid in so many ways? And part of the reason is that it makes it really easy to bring this data and pool it across sites and have thousands of scans, which has just been unheard of in the imaging community. Studies used to be like 20 people, and now they're 1,000 or 200 is like a minimum, you know? This is in a few years this has shifted. So why rest? It's easy. It means the data can be used for a long time, and it can be compared and pooled. And until somebody devises a paradigm that's easy to implement, it's a lot easier just to tell someone to keep their eyes open and hold still in the scanner. My honest sense is that this finding is this is the hope, right? That's where people are dreaming about this ending up. And the reality of it, I love that animation. The reality of it is that all of these categories that we define, these diagnostic categories, are probably much closer to the demographic information. And that that's going to be the criteria. DSM-5 is not incorporating imaging data into its diagnostic criteria, and until that happens in a diagnostic manual, this stuff will always be more predictable by the kinds of categories that are traditionally used. It's the same problems from other, I think genetics has dealt with, the same ethical dilemmas, and in terms of actual implementation, I don't think it's too early to tell, but at the same time, a path has been trodden for this before, and I think we can easily look to that to see where this is going to end up.